Ever look at a genetics question and wonder how you're supposed to magically know allele frequencies? That's where Hardy-Weinberg equilibrium comes in, and on the MCAT, it's less about memorization and more about recognizing patterns. In this video, we'll break down the key assumptions, walk through the famous p squared plus 2pq plus q squared equation, and show you how to quickly calculate genotype and allele frequencies without getting overwhelmed. By the end, you'll be able to spot these questions instantly and solve them with confidence. Hardy-Weinberg equilibrium describes a population in which allele and genotype frequencies remain constant across generations in the absence of evolutionary forces. This concept is foundational for MCAT genetics because it gives you a baseline expectation for what a non-evolving population looks like. If nothing is disturbing the gene pool, then the proportion of alleles, like dominant and recessive forms, does not change over time. That means evolution, defined as a change in allele frequencies, is not occurring. The equilibrium acts like a null hypothesis. If you observe deviations, you know something like natural selection or mutation is happening. On the MCAT, you are often asked to determine whether a population is in Hardy-Weinberg equilibrium or to calculate expected genotype frequencies under equilibrium conditions. A helpful way to think about it is like a perfectly balanced system where inheritance follows predictable probabilities generation after generation. This ties closely to Mendelian genetics, because Hardy-Weinberg assumes Mendelian inheritance patterns without outside interference. Real-world examples rarely meet all the criteria perfectly, but the model is still extremely useful. For example, geneticists can compare observed genotype frequencies in a population to expected Hardy-Weinberg frequencies to detect if a disease allele is under selection. A mnemonic to remember the idea is no evolution equals no change, which captures the essence of equilibrium. Understanding this concept sets the stage for everything else, including the equations and assumptions that define when Hardy-Weinberg applies. The Hardy-Weinberg equation p squared plus 2pq plus q squared equals 1 relates allele frequencies to genotype frequencies in a population. In this equation, p represents the frequency of the dominant allele, and q represents the frequency of the recessive allele, and together they must add up to 1. The terms p squared, 2pq, and q squared correspond to the genotype frequencies of homozygous dominant, heterozygous, and homozygous recessive individuals, respectively. On the MCAT, this equation is used constantly to calculate unknown frequencies, especially when given partial information, like the percentage of individuals expressing a recessive trait. A classic example is if 9% of a population shows a recessive phenotype, you know that equals q squared, so q is 0.3 and p must be 0.7. Then you can calculate heterozygotes using 2pq. A useful mnemonic is p squared is pure dominant, q squared is quiet, recessive, and 2pq is the mix in between. This equation assumes random mating and no evolutionary forces, so always check whether those conditions are stated or implied in a question. It also connects to probability rules because these genotype frequencies are essentially outcomes of allele combinations. Understanding how to quickly move between allele and genotype frequencies is a high-yield skill, and practicing mental math shortcuts can save time during the exam. The 5 assumptions of Hardy-Weinberg equilibrium are no mutation, random mating, no natural selection, extremely large population size, and no gene flow. These conditions are critical because they define when allele frequencies will remain constant. If even one assumption is violated, the population may evolve. For example, mutations introduce new alleles, natural selection favors certain traits, and gene flow migration brings alleles in or out of a population. Genetic drift, which occurs in small populations, can randomly change allele frequencies, which is why a very large population size is required. Random mating means individuals pair by chance rather than by genotype or phenotype, avoiding assortative mating effects. A common mnemonic to remember these is NO-MR, SG, standing for mutation, random mating, selection, and gene flow, with large population implied. On the MCAT, you may be asked which assumption is being violated in a scenario, such as a population bottleneck or selective pressure from antibiotics. Understanding these assumptions also connects to evolution topics like genetic drift and founder effects. For instance, a bottleneck reduces population size, violating the large population assumption and increasing drift. Recognizing these violations helps you quickly interpret passage-based questions and determine why observed data deviate from expected Hardy-Weinberg proportions. You can calculate allele frequencies from genotype frequencies and vice versa using Hardy-Weinberg relationships. This skill is essential for solving MCAT problems efficiently. If you are given genotype frequencies, such as the proportion of homozygous dominant, heterozygous, and homozygous recessive individuals, you can calculate allele frequencies by counting contributions of each genotype. For example, each homozygous dominant individual contributes 2 dominant alleles, while each heterozygote contributes 1. Mathematically, P equals the frequency of homozygous dominant plus half the frequency of heterozygotes. Similarly, Q equals the frequency of homozygous recessive plus half the heterozygotes. Once you have P and Q, you can plug them back into the Hardy-Weinberg equation to predict genotype distributions. On the MCAT, a common trick is that they give you genotype counts instead of frequencies, so you must convert counts into proportions before applying formulas. Another frequent scenario is being given only the recessive phenotype frequency, which corresponds to Q squared, and then working backward. This concept ties into probability and statistics, especially weighted averages. A helpful way to remember is alleles come from genotypes, meaning you break down genotypes into their allele contributions. Practicing this conversion process helps you move quickly between different types of genetic data. Deviations from Hardy-Weinberg equilibrium indicate that evolutionary forces such as selection, drift, mutation, or migration are acting on a population. This idea is crucial because Hardy-Weinberg is not just about calculations. It is a diagnostic tool. When observed genotype frequencies differ from expected values, something is influencing the population's genetics. For example, if there are fewer heterozygotes than expected, it may suggest inbreeding or assortative mating. If a particular allele becomes more common over time, natural selection may be favoring it. Genetic drift can cause random fluctuations, especially in small populations, while gene flow can introduce new alleles from neighboring populations. On the MCAT, you might see experimental data comparing expected and observed frequencies and be asked to explain the discrepancy. A classic example is antibiotic resistance in bacteria, where resistant alleles increase due to strong selective pressure. This concept also connects to evolution and population genetics more broadly, reinforcing the idea that evolution is simply a change in allele frequencies over time. A useful way to think about it is that Hardy-Weinberg gives you the expected snapshot, and any deviation tells a story about what forces are at play. Being able to interpret these deviations quickly can help you answer higher-level reasoning questions on the exam.